panelist is Dr. Samuel Barone, who is the Chief Medical Officer at Nanoscope Therapeutics, an optogenetic company addressing vision loss from advanced retinal degenerations. He is a board-certified ophthalmologist and vitro retinal surgeon with over 15 years of clinical development experience, including almost 5 years at the FDA as a reviewer of cell and gene therapy product submissions. Dr. Barone is going to talk about vision gains in Nanoscope's MCO 010 program and their work preparing for a commercial licensing application with the FDA. Welcome and thank you.
Great, thank you. I'm Sam Barone, I'm the Chief Medical Officer at Nanoscope Therapeutics. And yes, I have the dubious pleasure of standing between you and lunch. So I will try to be efficient and make it worth your while. Nanoscope Therapeutics, we are a leader in addressing severe vision loss from advanced retinal degenerations. And I'm happy to be able to share our work with you today. Amongst the other researchers who are here, the supporters of the community, but most importantly, the patients and their families. So for the fourth time today, this is how we see light comes into the front of the eye, hits the photoreceptors. It gets turned into an electrical signal that then gets through the multiple layers of the retina and to the brain. So that's how we perceive. In advanced retinal degenerations, you have loss of the photoreceptors, and that's why people lose vision. In the advanced forms, the photoreceptors are almost completely gone, and we have severe vision loss. And when we think about some of the programs and some of the therapies that are currently being evaluated to treat inherited retinal degenerations, they kind of fall into sort of two buckets. You have their gene replacement therapies, and other neuroprotective or gene editing elements that are trying to protect against further photoreceptor loss. In contrast to the things we've heard about this morning, we've heard about optogenetics, we've heard about the photo switches, which are targeting these advanced vision loss because there's already been the structural damage, it's already been set up, and all of those other those other programs and other mechanisms of action, they are going to be very limited to being able to improve vision once you have the structural damage, once you have the photoreceptor loss. I just wanted to emphasize that point. I think it was clear through the other presenters, but just wanted to re-emphasize that point. So what optogenetics is, it's with a single administration that's done in the office, intravitreal. As we heard here, you don't have to go to the operating room. And it targets those remaining cells that are in the inner retina and turning them into de facto photoreceptors. So it allows them to be photosensitive. I actually have a video here that...
A healthy retina is composed of an outer layer of rod and cone photoreceptors that convert light into electrical impulses, and an inner layer of ganglion cells that transmit these impulses to the brain. Between these layers are bipolar cells, which serve as the main pathway connecting the photoreceptors to the ganglion cells. In patients with inherited retinal diseases like retinitis pigmentosa or Stargardt disease, the rods and cones responsible for vision are damaged or missing. However, the bipolar and ganglion cells remain functional. MCO10 offers a groundbreaking solution. Administered as a single intravitreal injection, it utilizes an engineered adeno-associated virus to deliver the multi-characteristic opsin transgene directly to the retina. Using the mGlu-R6 promoter enhancer, the MCO transgene targets on bipolar cells, ensuring the opsin protein is precisely expressed in these cells. Once inside the bipolar cells, the opsin protein integrates into their membranes, transforming them into light-sensitive cells. This process, known as optogenetics, effectively reprograms the bipolar cells to detect light and generate electrical signals, essentially turning them into de facto photoreceptor cells. When ambient light hits these photosensitive bipolar cells, it generates an electrical impulse that is transferred to the retinal ganglion cells and sent to the brain. This creates an alternative pathway for vision to occur, even when rods and cones have been damaged due to retinal diseases.
Basically, Optogenetics aims to improve retinal sensitivity to light. And some of the things that you have also heard this morning, there's some differences between those different opsin proteins, those different light-sensitive proteins. Their ability to respond to different wavelengths of light into the different levels of light, the level of light around us. So you also saw that optogenetics has been going on for a while, and it's been through multiple programs, continues to evolve. And again, some of those opsin proteins allow for different ability to have a differentiation. So our opsin, it's sensitive at ambient levels of light. So that's like light that's around us in the room or outside. It doesn't require any additional goggles. It's also sensitive across multiple wavelengths of light, or across the whole sort of visible light spectrum. Will stimulate our opsin. And then additionally, what differentiates us is that we have been dosed our first patient in 2019. So we've been at this for almost 6 years in terms of dosing. And throughout this time, it continues to be safe. There have been no serious ocular adverse events in patients who have been dosed our MCO10 therapy. And then most importantly, or we can talk about our most advanced or latest stage therapy, or latest stage study, if you will, our Restore. The randomized controlled phase 2b/3 study. So what that means is that patients were randomly assigned to the active MCO10 dose group or to we call a sham control. So they went through a similar procedure similar to the administration procedure of just pressing a blunt syringe against the side of their eye. We followed the patients for 52 weeks to our primary endpoint, and then for another year. We call a masked fashion. So that means that the patients, their caregivers, the investigators, the staff at the clinical sites, even us, we didn't know what those patients, what treatment they got until after we analyzed the data, and we were able to look and see who got what and who didn't, and what were those differences in their response. Importantly, we saw, again, in contrast to these patients who are continuing to lose vision in this patient population, we actually saw a gain of vision. We saw greater than 0.3 logMAR improvements, which is the equivalent of 3 lines or 15 letters on a standard ETDRS eye chart. So again, patients gain vision in contrast to continuing to lose vision. And that 3 lines or 15 letters, that's the same threshold that's been used to support approval in multiple indications in ophthalmology, like in diabetic retinopathy and in macular degeneration. And then importantly, these vision improvements were what's called statistically significant compared to the control group. So that it wasn't by chance that we saw that these patients had better vision. These patients had severe vision loss, and in this trial, these patients had their vision loss was from advanced retinitis pigmentosa. And I can talk, I can say, hey, 3 lines, that's great. Be able to get approval for the FDA, or that's the same level of threshold. But what does that mean to you? What does that mean to patients in this room? And so that's why we actually have the words of one of the patients who participated in the study, so he can share, what kind of vision gains he experienced and how they were meaningful.
I started attending the testing at the center, and then they did the first treatment. And as we were driving, I began to tell her, I see a pin light coming in my eyes, and it's filling everything up with light. Do you see the cars? I said, I can see. I don't see details, but I see the cars and I see the houses. 15, 20 years since I've seen anything like this. And so she was surprised. I was surprised. The following week, I do rock climbing. As I got inside the gym, and the trainer was standing next to me, so as I got against the wall, and I said, oh my God. She said, what? I said, I can see the holes. I can see the colors of the hole. So she said, boy, you don't see the colors of the hole? I said, I typically don't. I'm feeling, but I don't see them. So she asked, what do you see? I can see red, orange, yellow, green, blue. I can see the colors, and they're vibrant, like real vibrant colors that I haven't seen since I was a kid.
So we thank Roy for sharing his experience, and importantly, these level of vision gains that we are seeing. We then, a big question that's come up or also comes up often in discussions like this is, well, what do the regulators think? Are these meaningful? Is this level of vision gain meaningful? And we're proud to say that we continue to be a leader and trailblaze in our discussions with the FDA. This has culminated in what's called a pre-BLA meeting, which is the last step before submitting a licensing application to the FDA, so that for approval, so that doctors can actually prescribe and administer the drug to real patients, not just to patients in a clinical trial. And importantly, they agreed with the contents of our package and have given us the green light to be able to support or to be able to submit that biologics license application. It's quite an endeavor. Now they're all electronic. They used to be paper and tens of thousands that would pages that would get delivered to the FDA by the truckload because they had to be in triplicate. But we will be announcing soon. Hope to be announcing soon that our initiation of that BLA submission. So we're really excited about that. And then another thing that's really important beyond you have your vision gains, very important. What do the regulators say? That's very important. But another thing that sort of, and these, thing that is beyond just regulatory approval is the access and the ability for patients to be able to get the drug. And so we've also led the way and trailblazed with in terms of access, market access. And there's an independent group called ICER, which is the Institute for Clinical and Economic Research. And they took a look at, there's a 9 to 10 month process that we had with them. They took a look, and then independently, then they provide recommendations to payers. It culminated in a public meeting at the end of the long process, and importantly, the panel agreed that this is something, there's an urgent medical need, this is something that is good for patients, and it is, there's economic value in the pricing model. So that's like a huge step for us to be able to, for payers to be able to agree, like, yeah, this is something that we should pay for because it's good for patients. So I'll wrap up here. Again, in summary, we are empowering patients and transforming lives. We have been providing vision gains to patients. They're clinically meaningful as viewed by both retina specialists and by the regulators, and payers are recognizing this as cost effective. MCO10 is effective, it's simple, it's safe. It's the first and only gene therapy that's shown the ability to restore meaningful vision in a randomized controlled study. Again, it's a one-time in-office intravitreal injection. And again, there's no genetic testing that is required because it is gene mutation agnostic. And there's been, across all of our studies, there has been no serious adverse events. So again, at Nanoscope, we're proud to be bringing back the light of hope. There are over 300 known genes and thousands of corresponding mutations for patients with inherited retinal degenerations. A gene-agnostic optogenetic technology platform makes a lot of sense. And again, we're proud to be leading the way. Thank you. There's a question in the back of the room.
Showed with the patient who explained what they could see after the procedure.
Yes.
Was that RP that they had, or was it choroideremia?
No, Roy had RP.
I'm sorry.
Roy has retinitis pigmentosa.
Yeah. So okay, so when this becomes available, would it be available for choroideremia, or do there have to be tests and things done again for strictly choroideremia?
Yeah, that's an excellent question. And so our initial work has been with retinitis pigmentosa. And unfortunately, there's a good chance because we've only done with retinitis pigmentosa that initially it will only be limited to patients with retinitis pigmentosa. But we see this as just opening the door. And our follow-on studies are going to have, when we show data and we show effectiveness with additional indications, we anticipate having being able to have a broad label claim that will be applicable to all inherited retinal diseases.
And what is the name of your company or your?
The company we're called Nanoscope Therapeutics. Thank you.
All right. Thank you again to all the speakers. And I do want to, as you all heard, these potential treatments are targeted towards advanced patients. The only reason I mention that is as we have speakers for the rest of the day and tomorrow and Saturday, there will be topics that are covering all age groups and many different types of research that's going on right now. So again, thank you very much. And of course, good luck to all three companies.